All right, so this is going to be the video to cover the last few slides of our first segment in microbiology. I wanted to, for one of you guys, one set of you to kind of, again, review this, and for the other class, we haven't talked about these bacteria. So the last classification of bacteria that we haven't covered, so we've covered proteobacteria, gram negatives, gram negative non-proteobacteria. Um, we've covered the mycobacteria, we've covered gram-positive bacteria, and so now we want to kind of, when we think about all these different kinds of bacteria, really what we're talking about are all the eubacteria. So, we covered a lot of these gram-positive, spirochetes, proteobacteria, cyanobacteria, plantomyces, so on and so forth. But when we talk about all these microorganisms, we talk about them because of their some aspect of their physiology, some aspect of their survival mechanics, which groups them together. Whether or not it's some part of their physiology, some part of their metabolism, some part of their uh, known environmental existence or niche space commonalities. But out of all of those things, one of the most interesting of all the different kinds of bacteria are called the deeply branching bacteria. And deeply branching bacteria simply means deeply branching into the branches of the phylogenetic tree. So when we talk about deeply branching bacteria, we're not talking about bacteria that have tendrils or that have roots or that can become filamentous. Because again, bacteria are finite in their shape because of their peptidoglycan and because of their cell wall. Now, the bacteria that most well describes the deeply branching bacteria that we've been able to identify or discover is a bacteria known as Deinococcus radiodurans, which you see here on the right. So this is Deinococcus radiodurans, which is Latin for bad grape. But the interesting thing about Deinococcus radiodurans is that it gets the species name because one thing that they found that it's resistant to is ionizing radiation. So there are different kinds of radiation. There's non-ionizing and there's ionizing radiation. And if we think about, the Earth from the very early stages of its existence and maybe the very early stages of life in totality, these type of microorganisms where the last universal common ancestor is most closely related would have experienced a lot of these same environmental stressors that Deinococcus radiodurans can survive in, and they're listed here, including ionizing radiation, because early on in the Earth's existence, there was no atmosphere, and no atmosphere means there's no protection against ultraviolet light and ionizing UV irradiation, extreme heat because of the existence of magma and the molten undercrust of the Earth before it had, cooled off. Drought and vacuum. Again, no atmosphere means there's nothing holding gases within inside the atmosphere of the Earth. So there would have been very little gravity. There would have been very little oxygen. There would have been very little existence. And so you would have been having high amounts if you have high amounts of heat, you have high amounts of drought because there's no water being able to be expelled onto the ground because it's so hot, it just gets evaporated immediately when it forms. And because there's a lot of carbon being formed in this time, right? Because magma and different types of carbon-based ash, you're getting lots and lots of acidity because again, uh, where there's not a lot of nitrogen and oxygen being captured in the environment, you're sending out a lot of carbon dioxide, carbon monoxide, and these can quickly become hydrogenated in a place where you have high rates of water evaporation, creating um, uh, carbonic acid. So microorganisms that can survive in a variety of different extreme environments are called polyextremophiles. And this makes these bacteria a little bit different than the archaea bacteria, because archaea bacteria are extremophiles because they typically exist in places that have a single stressor on their cell. So, for example, like halophiles. Halophiles are extreme salt uh environmental existing bacteria, meaning that they can live in places that have a salinity content which would be atypical for other organisms to live. So, for example, bacteria that live in the Dead Sea, where you won't find fish or cephalopods or anything else, you'll find these bacteria because even though the salt content is so ridiculously high, um, they thrive in that particular environment. But the point here is that that's an issue on osmosis and diffusion, right? Because of the extreme amounts of salt or solute in the environment in something like the Dead Sea. However, these bacteria can survive, but only in that singular uh, place, only in that place where there's a singular stressor on their cell physiology and cell ability to survival. Polyextremophiles are able to exist in a multitude of extremes because they are probably most closely related to the last universal common ancestor, which was exposed to all these different types of extreme conditions. So these bacteria that existed in the really early ages of the development of life and as the earth cooled most likely were responsible for basically eating one another. And we know that certain bacteria do eat other bacteria. And Vibrio, for example, is a good example of this, where a bacteria will eat or basically, uh, I guess, assimilate another microorganism into its cell cavity. When you have a bigger cell and a smaller cell, you generate an internal membrane, therefore creating what we know as the primitive endomembrane system. The idea is once these very primitive eukaryotes or prokaryotes that had early-ish endomembrane system by probably consuming and reconfiguring cells that they consumed, eventually we got cells that were so big that they were fundamentally bigger than bacteria. And so, and so you went from having two cells about the same size to kind of a uh how would you say, like an archaeal ancestor that probably had a endomembrane system, but a very primitive endomembrane system, but still no nucleus. So there's still no multiple chromosomes, there's still no nucleus, there's still no histones, right? But eventually, after these would grow large enough to continuously consume other bacteria, that eventually these would result in two segregated pieces of DNA and other development of endomembrane systems, including the nucleus, the rough and smooth ER, the Golgi apparatus, and most importantly, and directly related to the development of segregated pieces of DNA as it relates to forming other endomembrane systems would be the mitochondria. And this kind of makes sense when you consider where in the mitochondria ATP synthesis occurs. It's in the membrane of the matrix. And in it within those cisternae, there's a dual membrane, and then the dual membrane beyond that is the actual mitochondrial membrane. And that two membrane system is very reminiscent of the two membrane system you find in gram negatives, which probably would have been the more abundant of the bacteria at this time, as you were transitioning from uh, archaea bacteria to um, eubacteria. So in general, there are four classes of Archaea bacteria. They are the Archaeota, Archaeota, the Archaeota, and the Archaeota. So the Archaeota, these are basically there are two major classes of bacteria that fit in this or that are true representatives of this. There are methanogens and halobacteria. Um, Now, if you were playing the um, kind of the interim, we talked about the delta proteobacteria being kind of the first transition into eubacteria. The Archaeota are probably the bacteria most responsible for the generation of life. If you consider the two different types of families that are representative of this family, methanogens, which can produce methane, and halobacteria that exist within the places where you have large amounts of salt, you would find them to be here um, inside of the places where you find hydrothermal vents. Methanogens and especially bacteria that um, can produce methane. Methane is a carbon source, right? So if you're looking at here, the thermodynamics, if you have a carbon source, you can generate different carbon molecules. So methane is a hydrocarbon in a place where there's a lot of dissolved oxygen. So if you have dissolved oxygen in methane, you can quickly create hydrocarbons with oxygen from the dissolved oxygen, giving you generation of things like carbohydrates, amino acids, nucleic acids, or at least nucleosides, etc. So some aspect between the two of these, or within this phylum, the Archaeota are most likely going to be responsible to having a cell that is closely related to the last universal common ancestor, um, but on the branch, not deeply branching bacteria, but past the deeply branching bacteria. So evolved from there, but in a place where as the earth cooled and the new age of life that was not necessarily truly extreme or poly extremophile would have been your Archaeota. The Archaeota, which is the next group down, these are another group of bacteria that exist in volcanic springs and also can be found in the hydrothermal vents. And these bacteria basically serve the other half of this story because they have the ability to fix carbon. So they have the ability to fix carbon and also utilize inorganic molecules in combination to fixation of carbon to drive generation of different macromolecules and to drive metabolism. So because they exist in places that have extreme heat and have extreme amounts of sulfur, hydrothermal sulfur vents, and the ability to, again, fix carbon. So the ability to redox carbon molecules like carbon dioxide or dissolved oxygen, carbon, or places where you find inorganic sources of metabolic redox reactions like sulfur, phosphorus, etc. Being able to live in a place that has this in abundance, has the added environmental thermodynamics to have this in abundance leads to a situation where the Archaeota and the Archaeota probably coexisted in these deep sea hydrothermal vents in order to drive expression creation of giving off carbon-based molecules such as methane, and then fixing that carbon into carbon dioxide or carbonic acid, carboxylic acid, excuse me, that's required to make amino acids. The last two, the nanoarchaeota, There's only one species that's ever been discovered, the Nanoarchaeum equitans. These exist in hydrothermal vents, and you can find them in places like Yellowstone National Park. But these are obligate symbiotes, meaning that they need to exist um, symbiotically with an environment or a host. So, because they have this symbiosis, it means that they didn't exist prior to the thing that exists with their symbiosis. So, the likelihood that they were involved in really like uh the drivers of autotrophy and then eventually evolution into heterotrophy is probably unlikely. And then finally we have core archaeota. The core archaeota are really weird. We have not been able to cultivate them because they live in environments where cultivation is almost impossible to recreate, such as obsidian pools, molten lava places, so on and so forth. Now, when we talk about archaea bacteria and eubacteria, there are obviously differences based on where they exist. But one of the other really important kind of differences between the two is, the existence of what kind of phospholipids and how those phospholipids are created, especially as it relates to their ester linkages, um, and the ability and the outer membrane network, if you will, that has their S layer and their peptidoglycan layer. So in archaea bacteria, they have a different structure in their peptidoglycan layer that is not NAM and NAG. It is a different sugar moiety altogether, and they have a very simple kind of S layers. The S layer basically has these different kinds of proteins that still serve the same function as the S layer that we discussed previously. But this S layer is then sheathed by a peptidoglycan-like molecule, um, or series of molecules in order to protect the S layer. Whereas for for eubacteria, so all the other bacteria that are not archaea, the S layer extends beyond the cell membrane, so and cell wall. So here's the cell membrane, here's the cell wall. The S layer exists way out here. So if this was a gram-negative bacteria, this would be what the LPS is. For gram-positive, it goes well beyond where the cell wall exists, and again, because it's interacting with the environment. But the overall harshness of the environments that these extremophiles live in, the S layer needs to have the ability to not only, coordinate function within the cell, but also needs to more or less be protected so it doesn't become completely distorted or the protein becomes completely denatured because of the extreme environments that these bacteria live. The other really interesting thing is the side chain and ester linkage between or how the phospholipids are created in archaea bacteria versus eubacteria. So here's a phospholipid for archaea. Uh, it has a phenyl side chain and it has a singular carbon um, or carbon and oxygen ester linkage, whereas from eubacteria and in eukaryotes like us, we have a single carbon with a single oxygen, like you see here, but then there's a double bonded oxygen here. So we have a, a difference in side chain creation. And this results kind of uh, in a more flexible cell membrane. So for eukaryotes and prokaryotes, we have this kind of setup where we don't have contiguousness between the lipid tails. They actually have a distortion between between the outer leaflet and the inner leaflet. Whereas for archaea bacteria, because they are missing these other bonds within this ester linkage, they can drive, they can move other bonds along the tail length of the lipid group or the lipid moiety, and that creates a contiguousness between the tails, and then the tails can also interface with one another a little bit more readily. And this is probably due to the fact that because these cells exist in such extreme environments, the membrane has to work together with the sheath network and the S layer in order to protect things like the DNA and the general metabolic enzymes of the archaea bacteria to ensure that they can survive. So archaea bacteria also have a different kind of flagella. They have two really interesting kind of um, changes in two different kinds of molecules that you find in bacteria that are different in archaea. So the first is called archaeolum. So archaeolum is basically a type for pili structure. It is very similar. So here's archaeolum. Here's the type 4 pilus, right? So there is a lot of structural similarity between the two, and the archaeolum basically serves kind of as a um, rudimentary type 3 secretion system or a type 4 secretion system. So it has this needle-like projection. Um, and basically this serves kind of the same purpose as a type 4 or type 3 secretion system. It secretes molecules and effector molecules in DNA from one archaea to another. There is another really interesting structure that's found in a lot of different type of archaea bacteria, and that are these long thread-like projections. And these long thread-like projections are called cannuli. Now, cannuli can intermingle together to create these large, vast networks and webs that tether the bacteria to a environment that is, again, relatively harsh and probably fast moving. Now, the interesting thing is that at the end of these, or at the very terminal end of these molecules, you find something called the hami. The hami are basically these three pronged or three-pronged hooks at the end of the cannuli. So the cannuli look like this. They have these long extension, and then they have these three-pronged hooks at the end that serve as molecular anchors of the hami in different environments. So in eubacteria and most of the bacteria that we'll discuss, they have a very similar structure called fimbriae. Now fimbriae um, very much have um, have kind of a similar structure. Cannot do that. Sorry. Um, and so uh, fimbriae more or less look like this. If here's bacteria, these long polar extensions, and at the end of them, they have a hook or a tip. And it's the tip that actually binds to host proteins, host chemical moieties, et cetera, so on and so forth. Now, if you go back in the lecture real quick, back a wee bit, we talked about flagella. And if you look at the flagella also has a tip to it in both gram positives and in gram negatives. So one other function of flagella that is kind of universally functional, but also not really well thought of, is that because it has this tip molecule, which is fundamentally different than the flagellum that creates the tail, just like you have a tip protein with the hami and a tip at the end of the uh, fimbriae, which are these long, again, these long, sticky proteins, long adhesins. These tips are what drive the ability for the bacteria to adhere to certain things because they typically have amino acids which are extremely charged or otherwise have high rates of interfaces with other proteins, such as being a ring amino acid or something like that. So altogether, hami, fimbriae, and even flagella can all be used by a different kind of bacteria. Um, Archaea bacteria, the hami, flagella, and fimbriae for eubacteria as a extension beyond their cell membranes, beyond their cell walls, beyond their external decorations, and make for some of the very first contact cells make in their different environment or in different hosts. So if we're looking at kind of the overall kind of what things have and what things don't, right? Membranes that are lipids with branch hydrocarbons, um, only found in archaea, bacteria, and eukarya have linear hydrocarbons. And again, it has to do with the fact that the hydrocarbons that we have that double bonded oxygen, the carbonyl, if you will, between um, the phosphate head on the ester bond and to the lipid tail. And archaea bacteria do not. They have a contigularity, but that's because of that singular bond on either side of that ester bond. So the next bond out to the tail has a double bond, which branches the hydrocarbon, just like if you had an unsaturated fat. The chromosomes are circular. That is something that is maintained between the two different types of bacteria. They also lack a nuclear envelope and organelles because, again, they're both bacteria. However, here we go. Methionine as an initiator amino acid. Now we're talking about eukaryotes and archaea. So the rest of these are commonalities between eukaryotes and archaea, but not prokaryotes. No peptide glycan in the cell wall, because again, they have different sugar moieties. They're not NAM and NAG. It's just a different collection of molecular sugar moieties. Uh, Growth not inhibited by streptomycin or chloramphenicol because they target the ribosome, and therefore there's a difference in the ribosomes between archaea bacteria and eukarya versus prokaryotes. Histones associated with DNA, archaea bacteria have these. When we talked about earlier about the evolution or the origin of life, those very early archaea or deeply branching bacteria that became archaea most likely still did not have histones, but they eventually generated histone biology with their DNA. And here's another really important one, which is several types of RNA polymerase, which means that there's different kinds of RNAs that these bacteria can create, which are probably very similar in structure and function that the RNA is produced by the eukarya. All right, that's it. Hope you guys enjoyed, and let me know if you have any questions when you get back to class. Thanks.